Hi and Assalamualaikum. We are from Group 3 and today we are going to present about spectrophotometer. First off, this is our group member, which is we have first Imanina Izati, myself Anis Suraya, Hasha, Divya, Hani, and also my Hanisa.
A spectrophotometer is a scientific instrument used to measure the amount of light absorbed by a solution at a specific wavelength. It works by passing light through a sample and detecting how much of that light is absorbed by the molecules present. This absorbance is directly related to the concentration of the substance in the solution following the Beer-Lambert law. Spectrophotometers are commonly used in chemistry, biochemistry, environmental science, and medical diagnostics to analyze the concentration of compounds such as proteins, nucleic acids, and ions.
And next, we have the component of spectrophotometer. First, we have light source, coordinator, monochromator, wavelength selector, sample solution, detector, and digital display.
These are the functions of the components on the spectrophotometer. First, we have the light source. It is to provide the electromagnetic radiation. And then we have the sample holder, or we call it the cuvette. It is to hold the analyte. And then we have the display, or a computer. It is to show the data as spectra. And then we have the monochromator. It is to select specific wavelength used during the experiment. And then lastly, we have the detector. It is to measure the transmitted or the absorbed light.
Now, let's move on to the key principles behind spectrophotometer. Every molecule absorbs light at a specific wavelength. The instrument measures how much light is absorbed, and this absorbance is directly proportional to the concentration of the substance, which is known as a Beer-Lambert law. It's very important to select the correct wavelength to ensure both specificity and sensitivity in the measurement. Using monochromatic light, it means that the light of a single wavelength. It helps to increase the accuracy of the result. On the right side of the slide, we see the three main types of spectrophotometers, which is UV visible spectrometry, which detects electronic transition in molecules, and followingly infrared IR spectrophotometry, which measures molecular vibrations, and lastly atomic absorption spectrophotometry AAS, which is mainly used to analyze metal elements. These instruments are essential tools in many scientific fields, especially chemistry and biochemistry.
Moving on to the spectrophotometer numerical equation. The equation involved are the absorbance numerical equation. The first equation is the Beer's law, which telling that absorbance A is equals to the multiplication of molar extinction coefficient, concentration, and sample cuvette length, L, which is in centimeter. Second, there are two equations from the Lambert's law, which are first, absorbance A equals to negative log of transmittance T, or negative log of light passed through I over initial light intensity I naught. Second, absorbance A equals to 2 minus log of percentage of transmittance T. Next, the transmittance numerical equation. Transmittance means the fraction of incident light which is transmitted. First equation is transmittance is equal to light passed through I over initial light intensity I naught. Second, percentage of transmittance, which is the multiplication of transmittance itself with 100.
For industrial applications, spectrophotometer contribute the environment where it mostly used to measure of quantities of contaminants in the soil, water, and air. For instance, spectrophotometer help in detect of pollution like sulfur dioxide, nitrogen oxide, and particulate matter. This to ensure that quality of air and water meets legal requirements. Next, spectrophotometer highly depends to food and beverage to determine the color, texture, and composition of food products so it meets regulatory standard. For instance, spectrophotometer determine the concentration of sugar presence in fruit juice, protein presence in milk product, or presence of harmful compounds.
Next, let's take a look at its experimental usage. A spectrophotometer is used to determine the concentration of metal ions and organic solvents. It also helps us measure microbial growth and measure the concentration of biological molecules like DNA, RNA, or protein. Now, the procedure starts with preparing the sample. Then, we select the appropriate wavelength for the compound we are analyzing. After that, we calibrate the spectrophotometer using a blank solution and set the absorbance to 0. Once it's calibrated, we measure the absorbance of our sample by placing the cuvette with the sample in the spectrophotometer. If we are working with concentrations, we also construct a calibration curve to determine the concentration of an unknown solution. These steps help ensure accurate and reliable results in many types of laboratory experiments.
Lastly, in conclusion, the spectrophotometer is a powerful instrument for measuring light absorbance. It operates based on Beer-Lambert's law linking absorbance to concentration. It was widely used in scientific research, quality control, and also environmental testing. Its accuracy, speed, and versatility make the spectrophotometer an indispensable tool in academics and industrial laboratories. That's all from us. Thank you.